Okay, all right, so we found a mistake in the paragraph here. So we're going to mark out purebred because clearly if his fiance's wife or his fiance's nose reminds him of his mother, then it is not purebred because he has inherited an allele. Yeah, true. Okay, so what I want you to mark out on your paper there, yes, on the worksheet, I want you to actually mark out purebred. And I want you to replace it with the word hybrid. Okay? So we're going to replace that with the word hybrid. Okay, so what I want you to do is let's first figure out the genotypes of the aunt and the fiance. Okay? All right, so what is his aunt? She's round pants, stubby nose.
Little L, little L.
Okay, very good. Little s, little s, little L, little L. All right, now go ahead and write in the genotype for her fiance. And if you'll label those for me, which one is aunt and which one you're putting as the fiance. Okay, it's pure. We know that it's not going to be purebred long nose is heterozygous long nose, but purebred square pants. Okay. All right, so what would he be for purebred square pants? Okay, he's going to be big S, big L, little L. For nose. For nose, yeah. Okay, now, the next question says, what are the possible gamete combinations for each person? And what they mean by that is whenever meiosis is taking place and your cells are dividing out, and you've got, anaphase and telophase, you're making 2 new cells and then they'll split off again. You're going to have one of each of your alleles go to those gametes, to those sex cells. Because remember, in a human, how many chromosomes does a regular body cell or somatic cell have? It's going to have 46 in a regular body cell, but our gametes, our sex cells, will have 23. So they're going to you have 2 alleles for each trait. One of your alleles will go in one gamete. The other allele will go in another gamete. Okay, so it's they divide off so that you wind up getting one from each parent. And then when fertilization takes place, then that new person winds up with 2 from each parent, or one for each parent, so 2 for each trait. So the possible gamete combinations for each person, if from the aunt you are getting, you're going to get an allele each for round pants and stubby nose. So she only has one kind to give for body shape, and that's just a little S. That's it. So it doesn't matter. They can split off, but all she's ever going to give in her egg is a little S. That's all she's got. What about for her nose? A little L. That's it. So every one of her eggs are going to have little S's and little L's as far as the traits that she's passing on for the shape and her nose. Okay, so what about the fiance? What could his gametes be? All right, it could be a big S and a big L, or a big S and a little L. Okay, all he has are big S's to give. Okay, for square pants, but he has both a large L and a little L, so he could give either one of those in a gamete. Okay, all right. So then it asks, what are the possible genotypes for their children? Okay, this is where our large Punnett square comes into play. All right, so we're going to hang on excuse me, to that question. And I'll show you how we do it with the large Punnett square. Or do you think you can figure it out without using the Punnett square?
No.
Yes.
No.
Sure.
You think you can?
Why not?
All right, give it a shot. Look at it and see.
What?
Now we're gonna have a genotype of 4 letters. Okay. So what could be? What do we know they're getting from mom?
Little s.
We know they're getting a little s and a little l. Okay.
What?
Little s and a little l. What possibly could they get from
Big
dad?
S and a big L.
They could get a big s and a big l, or
A big S and a little L.
a big s. We know they got the little l from mom. So yeah, it could be either of those combinations.
I thought that since you didn't have to have.
All right, on number 7, it says, what are the genotypes of SpongeBob, who is heterozygous for his yellow body color and his square pants? So he's heterozygous for both. And his wife, Sponge Susie, who is blue and has round pants. Okay, so I want you to figure those out. So he's heterozygous for yellow body color. And he is heterozygous for square pants. Okay, so is that what you got for him? Big Y, big S, little s?
Yes.
Okay, now Sponge Susie is blue and she has round pants. Well, both of those are recessive traits,
That has to be lowercase.
so they're all going to be lowercase. Okay, now it's asking what are the possible gamete combinations for each person. Basically what you do is you take this first letter and you say, well, the first letter could be paired. Okay. The first letter could be paired with the first letter here, right? So it could be a big Y, big S. Or it could be paired with the second letter, a big Y, little s. Or he could give the little y, big S. Or
There's too many ors.
it give a little y, little s.
That's not written correctly.
It is written correctly. Remember, it is the capital letter, the dominant has to go in the front of each genotype. But for a dihybrid cross, you're looking at 2 different genotypes. Remember when we were looking at monohybrid, it was the same letter. You were doing an allele combination, the same letter, one from each parent. But in this is a gamete. So you're only giving one allele.
How do you know what to put in front?
You just, you're only giving one letter. It's not a regular genotype. On this genotype, you do dominant in front, lowercase and back on this genotype right here, okay? And you'll do the same thing for this. But this is a gamete. This is not a regular cell. A gamete only has one letter for each genotype. So this counts as its own little genotype. The Y does. S counts as its own little genotype. Okay? Because you're only given half. Because this is a sperm cell.
But how did you know that the Y went in front instead of the
We just used this. I mean, it doesn't matter.
It always knows matters.
It doesn't matter. You
Would
just
it be wrong if I said big S, little Y?
no.
Let it go.
You're just doing. We just did the traits in the order that they were given to us.
Okay.
So no, it doesn't matter. Okay, now Sponge Susie, what is the only gamete that she can make? She can only make a little y, little s. That's it. That's it. Okay. Now, when you're doing this dihybrid cross, you have to put each of these gametes in a box and then carry them across or down just like we did in this monohybrid cross that we did. So let me show you what I mean. All right, so we'll do her first, okay, since that's the easy one. In, whoops.
You're putting both of them in one bag.
Yes. In meiosis, remember you're getting half of the chromosomes you need in one cell, so we've got to combine these two to get a full set of chromosomes. So each parent's donating one. All right, so you have little y, little s, because that's all she can give. But now he can give big Y, big S. Okay, sorry. Got a message on my thing. It messed it up. Big Y, little s. Little y, big S. Or little y, little s. And it's because in meiosis, you make 4 different gametes. Okay, when you're at the end of meiosis, it goes through twice. You wind up with 4 cells at the end instead of 2 like in mitosis. Okay, then you do your Punnett square. Just like you did before, you bring your letters over and down, but keep the like letters together. Okay, so what I mean by that, this Y right here is going to go over. I put a capital Y in all of these. Okay, I'm going to have a little y that goes with it from mom. Okay, so I put whatever is above, I bring down in each square. I'm going to take my big S and go across right here. And then I'm going to take my little s's and go down.
So it ends up working out to where like it's supposed to, like they keep saying the little letters are not supposed to be in front of the big
Right.
It ends up working out to where they are.
Right. So that your genotypes
Correct.
are together and correct, yes. Okay, so you go ahead and you do that for all 16. On my second row, it is big Y, little y. I'm going to write this third row in a different color just because I can only write so small on this thing, so it all is kind of running together.
You say amen.
Amen.
When you're done, okay. All right. Yeah, it all runs together. It's because it's so small on my screen. So, all right. So it says, what is the chance of having a blue baby? Now, in order to have a blue baby, you're going to have 2 little Y's, right? So you have to go in every square here and count how many boxes have little Y's, both little Y's. Okay? Yeah, it just wants to know how many of these, what's the chance that you would have a blue baby, irregardless of the other stuff. It is going to be 50% because if you'll notice in these bottom 2 rows, every genotype is little y, little y. So that is 8 out of 16 boxes. So that is 50%. All right, so you have a 50% chance. So on your paper, I know you're doing it on your own paper. So if you will write blue and tell me that the chances of a blue baby are 8 out of 16. All right, now the next question asks, what's the chance of them being square pants? Now, what genotype would they have to be square pants? Okay, they could be big S, or big S, little s. That would still be square. So you're looking at anything that's big S, or big S, little s. All right, it is also going to be 50%, because if you'll look in this row and this row, all of them in that row are big S, little s. So that also is going to be the same thing, 8 out of 16 or 50%. All right, now the next question says, what's the chance of them being a blue square pants? So not only are they blue, little y, but they're also square pants, big S, or big S, little s.
25%.
How much?
25.
25 is correct. Because it is going to be 4 squares out of 16. Okay, and it would be 25%. So it would be 4 out of 16, 25%. Does everybody see how she got that? All right, and your last question on the worksheet says, what is the chance of a purebred recessive for both traits? Well, what do we mean by purebred recessive?
They have the same letter, but it's big and little.
All right, it's going to be, if it's purebred recessive means it's the little letters, but it's purebred. So it's all little letters. So it's going to be little y, little s, little s.
25.
And it's going to be 25%. It's that 4 out of 16 again, because if you look at this row, all of those are all little letters.
We're done.
Okay? Now, that is a very lengthy way to do this. There is a shorter way.
Oh, God.
Yay!
Of course there's a short
I guess
I want you to copy what it says there on this worksheet, where you see it says male, and then it has their,
chart thing.
yeah, their little chart. It's a,
The
uh-huh.
Can you talk about the one chart thing?
If
Or
you'll
you
look,
talk up here where it says,
I'm talking about right here at the bottom, this part.
or this.
Mm-hmm.
What are
This.
the L's above it?
Okay. So you're copying basically what I'm writing up here on the board. All right, now if you'll notice, these are 3 different sets of letters, 3 different genotypes. So what would you call this?
A tri.
A trihybrid, yes.
The tripod.
Does it say desired?
Desired, uh-huh. Okay, so if you will copy what I'm doing on the board up here, and I'm going to show you, we're going to work this out together. I'll show you how they worked it out, okay? So this is a tri-hybrid cross, and what they're saying, if they wanted to know what percentage or what the probability of offspring would be if they wanted big A, big B, big C, little c, okay, from these 2 parents. Now, we could do a huge tri-hybrid cross, but no. So that is 64 squares. Now, in your spare time, if you want to do that for fun, knock yourself out, okay? But
Extra credit problem.
yeah, that might be a great extra credit. It might be. But what we know is right here, it tells us these are the offspring that we're looking at. We want to know how many what's the chances, the likelihood of getting these three genotypes together in one individual, okay? So all you're looking at, they want to know big A, big A. Well, the dad can combine here. That's a big A, okay, with mom. Or if he gives this allele, this one could combine with that big A. So how many arrows did I just draw? Two. Okay? Now, if it goes the big A to the little a, that's not what we want. That's not what we're looking at. So we don't even have to do that. Okay? So in the male, the next one, you've got big B, little b. Well, that's what we want. We want big B, little b. Okay? So dad's big B could combine with mom's little b. His little b could combine with mom's big B. That's still big B, little b. Is there anything else? No, because big B with big B, that's not what we want. Little b with little b, no. Okay, so there's two arrows. Okay? All right. Then the same thing. You've got big C. This big C could combine with this little c. This big C could combine with this little c. Or this big C can combine with that little c. Or this one with this one. There's 4 arrows.
This is like a bunch of scrambled ones, though.
Now, it says put that in a fraction. There's 4 possibilities, okay? So on your fraction, you got 2 out of 4, which is 1/2, okay? What about the next one? 1/2. And the third one is 4 out of 4, which is 1. All right, so if you take that and you do 1/2 times 1/2 times 1, it equals 1/4. Remember, okay,
A half plus.
think a half times.
Calculator.
Okay. Yeah, 1/2 times 1/2 equals 1/4. Yeah. So, and you just multiply it by 1, but still going to be 1/4. Okay. So, there is a 1/4 chance or a 25% chance that if those two parents had children, that you would be able to get big A, big B, little b, big C, little c.
Small.
Lots easier than doing 64 square tri-hybrid cross. Just saying. Okay? So, on Monday, what we will do is we're going to practice doing some of these this way. Okay? So, if you will turn your work into me